Hello, everyone. Thank you for the invitation, and it's great to be here with you today. So I think I will transform you to a different space now, talking about spaces from the previous talk, although I don't know if it is going to be upstairs or downstairs, so you can think about it. But I work more on the applied side in looking at developing machine learning approaches for inferring and understanding cancer progression from large data sets. And I am associate professor at King's College London. My research is mostly machine learning, deep learning specialized, and we are interested in how we can improve treatment for cancer patients, but also how we can develop methodologies that help other areas. So the first talk, we talked about the Nobel Prize machine. I'm not very interested in that. As I said, we are interested in how we can improve cancer patient treatment, and we have 3 themes in the group. So first thing, we work on large imaging data sets where we interested how we can develop better deep learning approaches that can allow us to identify patterns from cellular imaging data that help pathologists or clinicians diagnose cancer more accurately, faster, and even decide what is the best treatment for the patient. From that, then the second theme in my group is to also explore using different data modalities. So for us, this is imaging, cellular imaging, microscopy, but also transcriptomics and genomics, and how we can use all these data to identify what things we can target in the patient, like genes or proteins, to improve their treatment. And finally, the third theme will combine the insights from the first 2 to help us design better drugs. So not only how we best use existing drugs, but also trying to think how we can design better drugs from the insights in the first 2 themes. And today I will be focusing on the first theme, which is how we can learn from large cellular imaging data. So to motivate our work, one thing we observe a lot in cancer imaging and in tumors is heterogeneity. So what you can see here is a tumor, and I hope you can see that there's green dots. Each dot is a single nuclei, but also you can see in the orange or red, but where the green is becoming orange, is the signaling of one of the cancer pathway, Wnt pathway, where you see there is a variability, how much it's expressed in different regions in the tumor. And this is where the dynamics become very important. We really struggle to get temporal data, but for us, is looking at this heterogeneity. We would be interested to understand the dynamics, how this heterogeneity can affect single cell behavior, how it can impact cancer patient outcome, and how they will respond to treatment. If you treat a patient that have different processes in different regions in the tumor, then it's likely this is going to have an impact on how well they will respond to treatment. So we worry a lot about the dynamics, but we can't capture it. So we really need approaches to understand this heterogeneity, and that's what we are interested in, how we can better use heterogeneity and single cell data to learn about the dynamics of cancer systems. And today I will be focusing on 2 main methods that we have been using to learn about the dynamics. So we have looked at the graph neural networks to look at tumor heterogeneity, and it nicely has been introduced in the previous session. And then I will talk about other approaches that are more scalable. So graph neural networks can be problematic for us because we have very large networks. So we are interested to look at more scalable approaches such as weakly supervised learning. And the first example I will give is based on multiplexed imaging data. So what these images show is like a tumor is 1 millimeter square in size, and what we can do is stain different proteins in these tumors. So we can stain proteins associated with vascularization in endothelial cells, CD31. We can look at proteins associated with extracellular matrix. We can also look for various cell types, so the proteins associated with macrophages, CD68, CD8, as well as various signaling pathways. So for that, by doing that in the same tumor, the same tumor and across 60 markers, we can get a lot of information about what's happening in each single cell in a snapshot of time, and how this information happen spatially as well. So I'm not gonna get deep in the biology, but this is like a protocol that we use is cyclic immunofluorescence. So the tumor is stained with 3 markers, because this is what we can do, 3 to 4, washed away and then stained again. So this is how we can generate that many proteins. So this is a special technique that gives us a lot of data for different proteins. And first thing is we need to process this data so it can be converted to numerical data. So what we do in the field is we segment each cell, detect the border of it, classify different cell types like immune cells, CD8 cells here, and then extract various features. Features include like the morphology, the elongation or the roundness of the object, the context of each cell, what is surrounding it, what is the surrounding cells, and the signaling. So what is the different proteins that are expressed. And we heard about very little data, people saying we have very little data. We don't have this issue here. So what we end up from each image is 3,000 cells. We have for this data set 746 tumors. So this is 3 million cells. So we have a huge amount of data, and we really need to make sense of it and try to come up with the principles underlying what's happening, what the different routes for cancer progression, and how we can design treatments for these different types of patients. So one thing we can look at is using clustering. We can visualize the different features. So I said like we have 60 markers. We can visualize these in the t-SNE space or reduced dimension, and we can identify different group of cells in the tumors. So we can see like in the clusters with the red stars indicate cells that are, or phenotypes associated with worse outcome, while the ones highlighted by a blue star associated with a better outcome. And one thing we know that sometimes as a cancer progresses, there is certain type of cells are depleted, so reduced, while more aggressive or transformed cells are increased. So we can also look how these different phenotypes are changed across different cancer stages. And we see that sometimes that clusters or phenotypes associated with worse outcome, which indicate here by red star, will increase with the cancer stage, and the other way around. So the ones that associated with better outcome are depleted or decreased as cancer progresses. But this is not always the case, so this is depending on the phenotype. What we also observe that if we look at the proportion of these different clusters or phenotypes in the patients, some of them are really rare. Like you will find less than 5% of the cells have this phenotype. But we know from the analysis that actually cluster 4 and 7 are associated with worse outcome. So despite being a rare event in the tumor, they can still have a significant impact. And this is why we are very interested in using this technology, because it's giving us this single cell resolution that allow us to understand better how this impact the tumor and how they can play a role in driving cancer patient outcomes. So I just use hierarchical clustering for this figure. And we can look at different pathways associated with these clusters. So again, I'm not going to get into details. We can look at the average signal. We can see what certain clusters associated, like DNA damage proteins, are reduced. We can also look at the correlation between these different clusters with immune cells, because we know that immune cells interact with the tumor and try to eliminate it. So here we can see nicely that clusters associated with better outcomes highlighted in blue have an increased proportion of immune cells, while clusters highlighted in red here have reduction of immune cells, which is here based on this normalized scale, except for CD68, which is associated with macrophages, and we know that it plays a role in protecting cancer cells. But what I mentioned is looking only at the single cells by themselves. If we start visualizing these different phenotypes in the tumor, and here I'm just highlighting the cancer cells, so the surrounding cells are stromal and immune cells, what you can see that there is a mixture of phenotypes. So each tumor will be not a single cell phenotype, but rather a mixture of components. And what we miss when we look at the single cells alone, that actually they also have a spatial dimension, how they are arranged around each other. So we see here the pink cluster and the green cluster of cells, while here the cells are from different phenotypes are intermingled with each other. So this is quite interesting because the question is how this different mixture or combination of different phenotypes will impact patient outcome, can have an answer how we can also treat patients. So what we can do is take for each patient and start looking how the proportion of each single cell phenotype in these tumors. So we can then cluster the tumors based on the mixture of phenotypes they have. And what we expect that actually most of these single cell phenotypes, if they are enriched in a certain cluster and they have a worse outcome, then the tumor also will have a worse outcome. But what is really interesting here is that we see, for example, that cluster 3 and 4 is when in tumor cluster 2 and 3, they have actually worse outcome. However, when a cluster 3 is not combined with the cluster 4, but with the cluster 1, the significance of the enrichment of these clusters in the tumor become less relevant, which is highlight the importance of looking not only what the tumor has of a certain phenotype, but also how these different phenotypes interact and impact each other and maybe support each other to drive cancer progression. Still, we didn't address the spatial component using these analyses. So one thing we can think about is actually the tumor in itself is an ecosystem, because we know these cells are interacting with each other, signaling to each other all the time. So we need better approaches to study these systems. And one way to study that is using graph neural networks. So as explained in the previous session, that graph neural networks can take an input as a network. And for us, this network, we represent the nodes as cells. And then for each cell, it's connected to its neighbor by an edge. And what's nice then that we can embed for each cell is various features. So we can embed the morphology and the signaling information. So I'm just gonna summarize again how these graph neural networks work without so much details. But initially with the cell network, we will have starting with the state of each cell, H. And then each cell will update its state based on its state as well as its neighbor states by multiplying with the adjacency matrix. And for us, we train the network to learn a certain outcome. So here for us, we were looking at the patient outcome and certain signature that I will not explain a lot here. But what we find that actually by doing that, we can learn the weight, which is very important because it starts telling us also about the importance of these different cells to the outcome we are looking at. And of course, once we done that, we were able to predict the outcome you are interested in with area under the ROC curve of 83%, which is really useful that we can predict a certain outcome. But what is very important to us is also to be able to explain our predictions, because this is where we can actually start gaining insight about the dynamics, but also what's happening in these patients. So what we can do is basically we can start looking at the weights learned by the network and use them to explain the network predictions. So what I'm showing here is highlighting for each cell what is the weight that were associated given the outcome. So the contribution of each cell to the outcome based on the graph neural networks. So the red indicate higher weight, so it is more relevant to the prediction, while blue indicate lower weight and lower contribution to the prediction. So what you see here, that certain region has been highlighted. Sometimes a lot of cells contribute to the outcome, and sometimes we see like a gradient of contributions. And for us, this is, we looked at these states of these cells and fed them back to the biologists to do experiments to validate some of these findings. So this is multiplex data, which is very complex and usually expensive to generate. There is another type of data called H&E, or histopathological data sets, and this is like usually used in the clinic. It highlights the nuclei and the basic structures in the tumors. So we were interested to see if this approach also can yield useful findings from just simpler data sets. So this is how this looks like. So we have tumors that are, in this project, we were looking at mesothelioma, which is a cancer that happens in the lining of the lung. And it's really bad. Once the patient has it, it's like almost a death sentence. So for these tumors, we have two main diagnoses, epithelioid and sarcomatoid, but also we have something in between called biphasic. And basically the biphasic is something, a mixture of those two phenotypes. So again, we explored the use of graph neural networks to see if it can help us to look at the heterogeneity and discover what are the association to the single cell level. So by this, we found that we can predict the outcome of the patient or the diagnosis very well. So we used two data cohorts, one for training, one for validation, and done the other way around. And we were achieving good area under the ROC curve. But again, as I mentioned, we are interested how we can then explain these predictions and use it to our advantage. And what you can see here, the same approach, by looking at the different cells associated with the outcome, we can identify the regions associated with the epithelioid phenotypes, and these were validated by the pathologist. So here the blue is associated with the epithelioid phenotype, while the red is associated with the sarcomatoid phenotype. So this is already a nice way that the pathologists can use to facilitate when they are looking at these images, which can be quite a tedious task. But then what we discussed about the biphasic, which is the most challenging, and actually the images or the patients where there is a lot of variability between pathologists. So what we can do then is highlight these regions for pathologists. So these are two biphasic tumors, and we can highlight where the regions that there is mixture of phenotypes, and this can help the pathologist make a better diagnosis of the patients. And here we did not tell the network what is an epithelioid cell or sarcomatoid cell again. We just told the network that actually this image is for epithelioid or sarcomatoid tumor. And the network itself discovered then the downstairs, which is a single cell phenotypes based on the large amount of data. And the take-home message from this is that graph neural networks can provide a very effective approach for characterizing tumor heterogeneity and identifying relevant subcellular populations, which can help us then start to hypothesize about the dynamics of the systems. Of course, something is really difficult to do in our work, that dynamics, because we have a patient and we have one snapshot of time, and we're trying to understand what happened in the future and try to predict what will happen. So this is where we start, like from one snapshot of time. So As I mentioned, graph neural networks are great, but there is challenges that they don't scale up well for larger data sets. So we have usually much larger images, and we started exploring weakly supervised learning approaches for that. So how this works is basically these images can be 10,000 of pixels by 10,000 of pixels. This is what the pathologist will look at, is quite large image. And because we can't feed all of this image into the deep learning models, what we do is we cut it in smaller pieces based on this grid. We then apply a feature extractor, so a network model that trained to learn useful features from these images, and then feed it to another deep learning model that can predict certain outcomes like diagnosis or response or whatever labels we are interested in. So weakly supervised learning is... Yeah, it is a typo, yes. So fully supervised learning when you have an object and it has a class. But when you have an image like this, it can be that the class, the abnormal region, it might be just this. It's not semi-supervised because this is like belong to the multiple instance, like multiple, it's like you have a bag of multiple objects and you need to predict based on the label of the bag, for example. You might have one abnormal object or one different class object in the bag or multiple ones, but you only have one label. So you don't have the label of the sub-objects or the cells here. We have a label for the entire image, and the entire image can contain normal regions, tumor regions, all of this. So that's why it's weakly supervised with A. Yeah. And what we do is basically try to predict the class without having any information from the pathologist. It's just like the large image label. So there is different approaches for that. One of them is vision transformers. This approach has been transformers applied very successfully to like ChatGPT, as you're aware. For images, how it works is that we again cut the image into small regions and feed them as a sequence to transformer encoder, which basically the main thing is it have multi-head attention that decide which regions in the image it should pay attention to, combine information from different locations, and then predict the class. Then there is another approach, which is consider that actually we have multiple regions that each region can have a label, but also the entire image has a label. So the each region, smaller region, is called instance classifier, and the entire bag here was called bag classifier. And what it tried to learn in parallel, what is the different instances in these images, what is the small image label as well as the whole image label by aggregating the information from the instance classifier. And one approach to do that is called dual stream multiple instance learning, DSMIL. So you can see here how it's combined different levels of information. This approach is much more scalable for our work, but also vision transformers have a specific power, so we've been testing these different methods. We also increased the complexity of this model called DSMIL Plus by adding another layer for learning features for the instance classifier. So what we found by applying this to a data set from glioma patients, we have 150 images from TCGA, so a publicly available data set. We have different diagnoses, 50 patients per each type of tumors. And then we tested these different approaches, including our model, DSMIL Plus. And we find that actually many of them perform very well. Our approach seemed to compare a little bit better, but it's also a much lower cost, computational cost. So this is a good way for us to test on larger data sets. But again, what we said, we are interested to explain why the model predicting that, because this is give us more information that first we can trust that the AI is learning the right things, and also give us hypotheses that we can feed back into our understanding. And here where I think that mathematical modeling can be great to validate some of these observations. So what we highlight here is the regions where the model things are more important. So in the darker red, you see the regions that are more important, while the blue is less important. And we see this for patients with different diagnoses. We also find that actually different models focus on different regions. So we have these different classes. We have each column here is corresponding to different model. And it seems the design of the model itself, it can affect how the model is learning. So maybe it's like us, we learn different ways. As the same for different deep learning models, they are learning different ways. So we're trying to work now with this to try to develop methods that can help us understand the dynamics and the heterogeneity of cancer patients, and how we can help the pathologists understand also what features they need to look at to assist them in their job. So in summary, I discussed why tumor heterogeneity is very important in order to design better therapies, because we now consider the complexity of the entire system. I also discussed about imaging, and from our side, we think that imaging is the most scalable approach, is very cost effective, and also discussed different deep learning approaches like graph networks and weakly supervised learning. So at the end, just conclude telling we work here in London. We also have very sunny, rainy days in following each other. So if you like the weather here, then come and join us. We have a very nice group, and we're looking for PhD and research staff. And finally, I would like to thank our funders, Welcome and MRC mainly, as well as the people who done the work. So Danny done the work on the weekly supervised learning. Fiona Genti is our collaboration from GE, who provided the multiplex data set. And Mark Eastwood, which is part of the PRISM team, we done the mesothelioma project. Thank you all for listening, and I'm happy to have questions.